Action. So, hello, my name is Shantele King and my partner,
My name is Toby Black.
and we did our project on the analysis of extracting genomic DNA from tropical fruits. A little background about us. We go to David H. Conant's Career Technology Center, and we are seniors in Joshua Wart's biotechnology class. This is our last semester. So let's get into it. So this is our introduction.
DNA is a molecule composed of chains that coil around each other to form a double helix carrying genetic instructions for the development, functioning, growth, and reproduction of all known organisms and many viruses. And we chose a fruit, which is a ripened ovary that has two basic parts, the flesh and the seeds. The seeds are the propagule the propagule, and have the presence of full complement of DNA in them, particularly the embryo, which allows for genetic continuity. DNA is also present in the flesh because there has to be DNA in each and every cell and degenerates when the flesh is consumed or decomposes.
Um, the basis of two complementary DNA strands from hydrogen bonds between adenine A and thymine T or guanine G and cytosine C give a DNA molecule its double phosphate group. The structural portion of the DNA molecule is composed of alternating sugar composed of beta D deoxyribose or phosphate groups. The research experiments will focus on analyzing the extraction of DNA from five different types of fruit: watermelon, kiwi, lemon, pomegranates, and seedless green grapes. Watermelon grows mostly in Texas, Georgia, and Florida, also California, and they are known to need long, warm seasons in order to grow well and take up to 90 days to be fully reopened, to become fully reopened. Kiwis are grown best in California, also New Zealand, and live in places with mild winters ranging from between 3 to 5 years to become fully grown. Lemons also take up to 3 years in order to become fully developed and grow in spaces such as Spain, Italy, Morocco, Greece, and Turkey. Um, Pomegranates grow
Um,
mostly in the Middle East, South Asia, and the Mediterranean region and take 5 to 7 months to become fully developed. Lastly, green grapes are grown from a vine and can take up to 3 years to produce um, viable grapes. They are grown on south or southwest facing walls or fences and are mostly produced in Italy. The objective of this research is to extract genomic DNA from tropical fruits. This research allows us to practice DNA extraction and gives us the opportunity to learn more about DNA from fruits. By doing this project, an accurate comparison of one fruit to the other is shown, allowing for an accurate hypothesis and an answer to the question.
our hypothesis in question for this project was figuring out which tropical fruit would have the most genomic DNA inside of it. And we hypothesized that kiwis will have the most genomic DNA because they have a hexaploid containing 6 chromosomes.
Which was more than all the other fruit. So this is our material list. We used um, basically microcentrifuge machine, sodium chloride, sodium sulfate, tris, ethanol to make our what do we use?
To make our lysis buffer
Yeah,
solution.
to make our so that we
Most of
could
the
which we use to separate the genomic DNA from the like gush stuff that we didn't like, the mush basically is what we called it. Um, we used a hot plate machine, a stir bar, the fruits, the lemon, kiwi, watermelon, pomegranate, grapes, Bunsen funnel system, which we use um, to
to
basically funnel
filter out
out,
each.
filter out, yeah, protease, beaker, flask, graduated cylinder, plastic spatula, tray, scalpel, blender machine, ice pad, thermal. thermometer, needle pipette, and weigh paper. So for
A total of
protocol,
a bag of grapes and one watermelon, 4 pomegranates, and 20 kiwis were peeled and cut using a scalpel to weigh up 250 grams in each of three different trials. After peeling all the fruits, 10 milliliters of tris, EDTA, SDS, and 12 milliliters of recycled NaCl were all added to 168 millimeters of DHCl in order to make the lysis buffer solution. 250 grams of each fruit were poured into the blender and blended for 20 seconds. The fruit was then poured into a 400 milliliter beaker and 100 milliliters of lysis buffer solution was added. The solution was placed on the stir-by machine and stirred at 500 rotates per minute while slowly reaching up into temperature to hit 60 degrees Celsius. Then the timer was put on for 15 minutes to track the Celsius or the
um,
temperature.
while we heated, while being heated at 60 degrees Celsius, 0.001 grams of protease was measured out and added to the beaker. The solution was strained while sitting on a cooling pad using a Bunsen funnel system. After draining all the solution, it was placed in the freezer for 30 minutes until below 0 degrees Celsius. 20 milliliters of each solution was added in 7 separate centrifuge tubes, and 10 milliliters of ethanol was inserted into each centrifuge tube to initiate genomic DNA extraction. This process was repeated for each fruit. The total amount of genomic DNA observed from each tube was recorded. Um,
And from each tube that was recorded, this was our results. Kiwi did not have the most. It ended up being watermelon with 17.3 milliliters, kiwi at 14.69 milliliters, lemon at 14.16, and pomegranate at 1.72. And grape did not have any.
um, application. Um, Fruit is an important factor of everyday life throughout the world. Many people eat fruit and plants from gardens and grocery stores every day. As the world continues to evolve, people continue to look for ways to make the world better. Looking into genomic DNA from fruits could help with changing the way fruits are grown to make them healthier and less likely to harm our body. Keeping chemicals and bugs out of fruits that we eat every day. Studies could also be made to try and change how long they are ripe and stay ripe. Many things can be looked into with the help of DNA, things that could help us live a healthier lifestyle.
And in conclusion, after completing the protocol, it was resulted that watermelon had the most genomic DNA with the average of 17.30 milliliters of genomic DNA extracted. Kiwi with 14.69, lemon with 14.16, and pomegranate with 1.72. And grapes did not contain any. As we said earlier, kiwi was hypothesized to have the most genomic DNA because of the hexaploid set of chromosomes. And our hypothesis was incorrect because watermelon had the most. Um, there were many experimental errors during our project. We didn't have enough time management for it, which was allowed to delay for each of the trials. So one trial would have like barely any, then the next trial would have the most, and the next trial would not have any at all. Which we found this probably um, corrupted the results a lot due to having the rush one and not the other.
At the end of this page. Um, this caused for corrupted results in the DNA, allowing one sample to contain a large amount of genomic DNA than other samples. Another experimental error was not having enough buffer solution. In the middle of the procedure, the blended fruits had to sit and wait till more buffer solution was made. Another experimental error was forgetting to add protease to one of the samples. This caused a lot of trouble having to redo the procedure for grapes 4 times instead of 3, causing a minor setback in time. In future studies, the mass would be collected from the fruit rather than the measurements in millimeters, which would give a more accurate result of the DNA. The group of fruit would also be looked into deeper when it comes to how well people have been able to extract DNA from it, making sure that all the fruits used have the ability for DNA extraction. It would also be good if vegetables were experimented on. Experimented. finding out if DNA can be extracted from vegetables as well as fruits.
And we
Um,
chose this project because we feel like it was a very important part of the fruit plays a very important part of the world, since almost everybody needs and a lot of people eat fruit. And I think it's really important to know what you're actually eating and consuming every day. And this is important because the good and bad which could be inside of your body from what you're eating.
it was also a great way for us to like um, really um, go farther when extracting DNA. We have in class extracted DNA from saliva, and it was honestly pretty. It was a cool project as well for us. So being able to extract it from fruits also allowed us to, expand our knowledge on extracting DNA. Like being able to do it ourselves and do the project together as a team was really really nice. So yeah.
And as you said earlier, it's not really normal you see DNA and fruits together in like the same sentence, which also was very enjoyable to be able to extract genomic DNA from other than something like from a person.
Yeah.
I feel like it was very different extracting this from fruit.
Yeah. And I feel like for it to be our first trial with doing DNA extraction from fruits, I think it print went pretty smoothly, even though we did have some things that we could work on, such as like time management. Um, we would we would like to in the future um, be able to manage how we did the project a little better rather than just going in head on. But other than that, it went pretty smoothly, and we did enjoy, doing the project and getting hands on and doing something different compared to the norm, I guess you could say. But yeah. So that is our project on the extraction of genomic DNA from tropical fruits. I hope that you did enjoy it and that you are staying safe and with everything that's going on, stuff that we couldn't be in person to meet hi. But I did enjoy doing the project and being able to work with my partner Toby. So uh, yeah, that's about it. You guys have a great rest of your day and stay safe out there.